Sorry, what's your name?
Sage.
Sage. Okay, I'm very happy that Sage mentioned the distinction between the Human Genome Project and the 1,000 Genomes Project. Because the Human Genome Project is trying to get the first genome for us, for the human species. And the 1,000 Genomes Project is trying to get the genome for many more individuals. And the purpose of getting more genomes is to identify difference across different genomes. And basically we're trying to find the sequence of the nucleotide in each genome and try to find a difference in those sequences. It's a very interesting perspective. My perspective about DNA is just a string of characters. So when in my daily research, I'm dealing with this kind of data. So actually, a lot of researchers in this field in computational genomics, some of them actually has they comes from physics and comes from computational biology and computational science. Some of the research that actually didn't has a deep background in biology, and sometimes they don't really need much background in biology because for them they can treat DNA just as information, as a string of letters, and you'll be surprised how much information you can learn just from those information alone. To start with, I want to reiterate some information from the human reference genome, and this is already covered in our first lecture. The human reference genome was generated by this specific project. It's an international collaborative effort, officially started in 1990, and it takes about 15 years to finish. But the ideas goes back to 1984. It was finished in 2003. And the thing is that when they announced that the project was finished, they didn't actually get all the sequence in the genome. They only get about 90% of the genome. And that's because some part of the genome is very complicated, a lot of repetitive sequence, and especially in the centromere region, it's just very difficult to figure out what are the sequence of nucleotides in those regions. So it was only 90% complete in 2003, and there's another consortium, the Genome Reference Consortium, that actually continue to improve the human reference genome. And one side note is that for most of the slides, I try to put some basic concept at the bottom of the slides with a specific star. Those are some basic concepts you need to know. And we say sequencing a lot. Sequencing basically is the process to determine the sequence of nucleotide in DNA. And here are different versions of the human genome. When it was first released in about 2001, it was the human genome version 8. Over the years, it has been improved. Nowadays, the Genome Reference Consortium was founded in 2008, and it released the first major release. It's the human genome version 19, and now the version we have is version 38. And each time they release a new version, they make a lot of fixes and they add some new information to the version. So the human reference genome until now is still not considered as 100% coverage. Each time it released, it was. So this is the information about the most recent release, the version 38. And here it has three kinds of improvement. The first kind, as indicated by the triangle, are the genomic regions. They are very complicated. It could not be. It could not be represented by a single one type of sequence. So you need to generate an alternative sequence as reference, because basically it's too different among individuals. So they generate additional reference. And there are also places that were errors from previous assembly. They just fix the error, and they also increase some new places. Previously, they was considered as gap. using new technology, they were able to fill in those gaps. Just remember that the human genome is still a work of improvement in process. So one major task of the Human Genome Consortium is size DNA sequencing. So the DNA sequencing is the process to determine the sequence of nucleotide in DNA. But another major task is to annotate those sequences. You have those letters, but what does those letters mean? So this is genome annotation. Genome annotation is basically trying to make sense of the genome to determine where are the sequences that encode protein, where are the sequences that regulate the expression of those genes. So basically it's the process to identify genes and other features in the genome, for example, repeat, insertion deletion from some transposon elements. And so originally it was considered as the most important aspect of the human genome is the coding sequence, the sequence that translate into protein. And you can see that only 1% of the human genome is actually translating into protein sequence. The other 99%, what do they do? At the very beginning, they were considered as junk DNA. They think that they didn't do much. But nowadays, with more research, we actually starting to get to understand what those junk DNA do. One major function is the regulation of gene expression. So this is a basic structure of the gene. We have the exons. So exons and introns. Exons are those that are transcribed into mRNA. Introns was retained, it was not transcribed. And in the mRNA, only part of it is translated into protein, and this is the coding regions for CS. And the two untranscribed regions, 5' and 3' UTR. And the interesting thing is that when you look at the sequence level of mRNA and DNA, for example, at this place, at the junction of exon and intron, they usually have specific nucleotide. And also this is the starting codon for the translation. So this, and for all transcript, you need to have a stop codon. And all those specific features of a transcript, those features can be used to predict, like in a DNA sequence, where is a gene. So for in the Human Genome Project, when they do genome annotation, one major approach is to make prediction based on the knowledge we have. So based on this kind of knowledge, and go through the DNA sequence to see where we have this kind of specific sequence, and that help us annotate the genome and help us identify coding sequence. 99% of the genome is considered as non-coding sequence. You can it includes the introns and the UTRs in the protein coding region. So only part they intersperse with exons. Many other non-coding RNA, for example, transfer RNA, ribosomal RNA, and microRNAs. Those are all involved. So the microRNA and the long non-coding RNA, those are involved in regulating expression of some protein coding genes. There are a huge number of pseudogenes. So if you look at the number for protein coding gene, it's about 20,000. There are 13,000 genes. So pseudogenes basically are just copies of protein coding genes. Sometimes in evolution, a specific protein coding gene duplicated, so you have 2 copies of those genes. One of the copy accumulated mutation, so it was no longer active, and it turned into a pseudogene. And there are a huge number of them in the human genome. And there are many other non-coding sequence, regulatory sequence, promoter enhancer, and there are many transposons and repetitive sequence in the genome. So when we look at the genome, this is the genome, and if we pick one of the specific chromosomes, for example, pick a chromosome 18 here, we can, this figure gives us an idea of where the density of the gene and the density of some non-coding genes on this chromosome. And the first thing is that if you look at the centromere, there's no protein coding gene in that specific region. And the problem is that basically we don't have much information. We don't have the sequence for this region. There's one explanation, or something we already know because we don't have much information for the centromere. The other things that for the centromere, indeed it was mainly repetitive sequence. So there's not many functional elements there. And also for the centromere region, it is usually high in GC for the two specific type of nucleotide, G and C. They tend to have very high content in the centromere region, and that's another feature of the centromere. And if you zoom in to a specific region on the chromosome, and here are the annotation of that specific region. Basically, it's just different structure of the gene. And sometimes you can see that, for example, it has the same name of the gene, but it has at the same position. But it has different. So the thicker line is the axon, and in between those axons are intron sequence, and these are thus different transcript of the same gene. And all these are the information you get from the human reference genome. But the human reference genome is only one representative genome, and one important note is that it is actually a haploid genome. We humans are diploid, and they only have information about one of the haploid genome. One important question is that if we get the genome from two individuals, how many differences can we find? And where are those differences? Do they have any functional potentials? So those are the major questions we can address with sequencing more human genomes, and that's what we're going to talk about today, about genetic variations. And some basic concepts, what are the different types of genetic variation? The simplest type is single nucleotide variant, sometimes called single nucleotide polymorphisms. Those are the difference at a single nucleotide addition. So for example, this is a change from AT pair to a GC pair, and that's a change at one base pair position. This is considered as a single nucleotide change. And the difference between single nucleotide variant and SNP is that, strictly speaking, polymorph for a SNP to be a polymorphism, you need to reach a frequency of more than 1% in a specific human populations. But nowadays, when people see a difference at one base pair position, they just call it SNP. So they didn't really follow this strict requirement. And another important concept is allele. So we at the very beginning, when we say allele, we refer to different versions of the same gene. But in the context of a SNP, or in a context of genetic variant, different possible nucleotide is also considered as an allele. So for example, for this specific position, it could have the A allele or the G allele. So either one of them is considered as an allele. For most of the SNP, they are biallelic. Basically, they have only two possible alleles in this case. But sometimes you can have multi-allelic SNP. Basically, you have more than two alleles for this specific position. In this case, is 3. But the maximum number is just 4 because there are 4 possible nucleotides. That's about a single nucleotide variant. Another type of very common genetic variation are the insertion-deletion polymorphism. And here I emphasize small insertion-deletion polymorphism. We call them indel. And this is a very important class of genetic variation that we study. And basically, it's just for example, this is a wild type sequence, and in this allele, so these are 3 different allele can refer to a specific genomic region. It could be more than 1 base pair. So we can call this as 3 possible allele in the same position. And this allele, this is the wild type allele. So this allele has a 3 base pair deletion, and this allele has 4 base pair insertion. And this kind of difference is called insertion-deletion polymorphism. And it's so for some of the insertion-deletion, it can involve very, very long strands of nucleotide, for example, much more than 50 base pair. But for most of the recent research, they tend to focus on small indel, and it is mainly a limitation of the technology we have right now. For the sequencing technology, it's easy for us to identify small insertion deletion. It's very difficult for the current sequencing technology to identify deletions that's very long. And for the next lecture, when I talk about the sequencing technology, I can explain why it is so difficult to find long insertion and deletions. So the last major category of genetic variation is structural variation. Basically, you lump everything besides those 2 into structural variation. So they include very large insertion and deletion, and also reversions. Insertion. So when we say insertion deletion, we always say that by comparing to a reference genome. So when we get the reference genome, we compare a new genome to a reference genome. And this, for example, this genome is missing this whole trunk. So we call this allele as a deletion. And for this one, it has an insertion compared to the reference genome. So insertion deletion is relative. And some of the insertion is mediated by mobile element insertion. So transposon repetitive element could be a result of insertion. And pendant duplication, you can consider that as an inversion. And copy number variation. Copy number variation sometimes is the same thing as repeat, depends on the specific context. So these are the major type of genetic variation, and now we can go into this paper and see what we find about genetic variation when we sequence 1,000 genomes. So this is the assigned reading for this week. It's a publication from the 1,000 Genomes Project. So as the name indicates, the purpose of this project is to sequence the genome, to sequence 1,000 genomes. So this project was started in 2008. It was a few years after the Human Genome Project was finished. The original goal was to sequence at least 1,000 individuals, but it ended up, when it finished in 2013, it sequenced many more, 2,500 individuals. And the reason that this project exceeds the expectation was that the sequencing technology developed so fast in those few years that it significantly reduced the difficulty and the cost to get that large amount of genome. this project, similar to the Human Genomes Project, it was an international effort. Most of the institutes that participated in the Human Genome Sequencing project, they also participated in this project. And by sequencing a large number of individuals, the goal was to identify genetic variants with frequency of at least 1%. Because think about that, if we have 1,000 genomes, a frequency of 1% indicate that that mutation or that difference is only found in about 10, 1% is about found in 10 individuals. So if we want to get even like genetic variants at lower frequency, very rare mutation, it's just very difficult to find them. We need to get many more individuals. And about this, the samples for this project, they collect samples from 26 population, regional population. And here are the locations of those samples. They collect samples from different continental regions. They try to have a similar amount from different continents. They have 5 from East Asia. They have 5 from South Asia, 5 from Europeans, then 7 from Africa, and then 4 from America. And the thing is that those samples are colored by their ancestry. So for example, for samples that has African ancestry, they color in yellow. And you can see that actually a few samples that color in yellow is actually collected in America. So basically those are individuals with African ancestry that live in America. So they just, for the ease of the project, they just collect sample from in America, but those samples are actually American. And similarly, actually one of the European sample was actually collected in Utah. So when you look at, so this is the list of the sample, and this is the specific sample I was talking about. This is a European sample, but it was actually collected in Utah. And a lot of research work has been done in this specific sample. Each sample has about 100 individuals, so they try to get similar amount for each local sample. And one interesting thing to notice about the American sample, so the American sample highlighted in red here, they are considered, they are individuals that has a mixed ancestry. Basically, those population, they have no history of admixture between populations of different ancestry. For example, the Native American, the Europeans, and Africans. So the sample collected in America, so those samples are likely to have ancestry from Europeans, Africans, and Native Americans. And by comparing to those samples collected from Europe, like at least in the recent history, they don't have a strong admixture with a different population. And those different history can actually has a strong impact on the pattern of genetic variation we are going to observe. And the technology used in the 1,000 Genomes Project, and as I mentioned, I'm going to talk. We are going to focus on the sequencing technology in the next lecture. Here, I just want to give you an overview, just to touch a little bit about this topic. And this is going to be the case throughout the lecture I'm going to give, is that because we are focusing on a specific paper, and each paper, because they are the most advanced research we have right now, and each research is going to touch on different topics that are relevant to human genetics. And for us, each lecture, we try to focus on a specific topic, but we are trying to cover a little bit about slightly a different topic in this lecture. And so for the sequencing technology used in this specific project, so I say sequencing technology, it implies that we are going to read this DNA sequence. But sometimes you can use the sequencing technology to read only one place. You don't need to read the whole sequence. You just read one specific place. And it's called targeted sequencing or genotyping. So basically, you have one specific position that you are interested in. For example, a position that based on previous study, we already know that on a specific chromosome, for example, chromosome 1 at about 1 million, at that specific position, we know that that position has a very important genetic variant, and we want to know for specific individuals what's the genotype at this specific position. You can do a targeted sequencing and get to know the genotype for this specific position. So this technology is called genotyping. It's genotyping by doing a sequencing. And there are other different technology you can do genotyping. It's called microarray. We're going to talk about it next lecture. So for whole genome sequencing, so this for the 1,000 Genomes project has it used 4 technologies. The first one is whole genome sequencing. Basically, this technology tries to read the sequence of the whole genome. And so this is the ideal technology. Ideally, we want to apply this technology for all the sequence we are going to study. But the thing is that whole genome sequencing at that time was really still quite expensive. So you could not afford sequencing that many genomes. And basically, so you can see that they only do whole genome sequencing to a low depth. Depth is a concept used in sequencing technology. Basically, each one depth means that you read each position once. If you have a 7 depth, it means that you try to read each position 7 times. But the thing is that when you try to read the genome 7 times, the distribution is not even. Some place you can read more than 10 times. Some place you only read once. So that's but this is the average. The second technology used in this project is the exome sequencing technology. So exome is the collection of all exons in the genome. So exons are the sequence that turn into protein sequence. And remember that in exome, not all exons are turned into protein. They are still untranslated regions, UTRs. And for whole exome sequencing, basically you can only focus on these specific regions. And do you still remember how much does exome account for the whole genome? 1%, right? So when you focus on whole exome, you significantly reduce the sequencing cost. And the idea is that 1% of the genome generate all the protein, all the enzymes that function in our body. So that 1% of the genome is the most important. So we can focus on this most important region. So those are the 3 technology, and these are the description of each technology. And ideally, if you want to get a high quality genome, you need to get a 30 plus coverage. So you can cover everything in the genome. In that case, you can identify all genetic variations in the genome. For the whole exome sequencing, because it's much less expensive, you can actually sequence to very, very high depth. But it's very cost effective. You can also, for the regions that have coverage, you can find all the genetic variants, but it's just 1% of the genome. The targeted sequencing, similarly, it further reduces the cost by focusing on specific position. So it's most cost effective, but you only get to know variants at the positions that you are interested in. Question?
For the whole exome sequencing, is it to figure out what the exome is? Are you using a reference genome, or is the technology figuring out where the exome is?
That's a great question. So the question is that, so actually the question applies to whole genome sequencing or exome sequencing. Basically, after you get the sequencing result from some machine, how do you figure out if there's genetic variant at specific position, right? So actually, something I'm going to talk about right here. So is so in the human genome project, we were trying to get the first genome for a human. At that time, we were trying to assemble the genome. So we don't have anything we can compare to. We just we try to piece together to view the first human genome. This is the process known as genome assembly. For all species that don't have genome, you need to do a genome assembly first, get the first genome. The idea is that you get the DNA from some participant, you process them, you send them to a sequencing machine. The data you're going to get from a sequencing machine are fragments of DNA, fragments of sequence. And you try to piece them together and try to build the sequence that correspond to the original genome. So this is the process of building a genome assembly. Now, in the 1,000 Genomes Project, we already have our first human reference genome. We don't go. So theoretically, we can do the same thing. We get the sequencing result for each genome. We can go through this process, assemble the genome for each individual, but this is very challenging. With a reference genome, it's much easier for us to find genetic variants. What we need to do is that, so those are the sequencing results from the sequencing machine. We just compare them to the reference genome. And the idea is that for any two genomes, there are only small difference. It's unlikely to have more than 2 or 3 differences in one sequencing fragment. So you can compare that fragment across the genome, it allow 1 or 2 differences. And then you can place that sequencing fragment to a reference genome. So we know that, okay, that fragment comes from here. And when you put them all together, you're kind of building a genome for the new sample. And by looking at the positions that are different in your sequencing result and in the reference genome, this actually is a genetic variation. So this is the difference between the new genome and the reference genome. So this is the process. We identify genetic variant. No matter what kind of sequencing you do, whole genome sequencing or exome sequencing or targeted sequencing, after you get the fragment, you compare them to reference. And then you find the positions that are different from the reference genome. And this is the process known as variant discovery or genotype calling. Basically trying to call the genotype for the new sample. And for this specific individuals, so remember that when we do sequencing, when we get DNA from any individual, they are diploid. So you actually have the information about both of the chromosomes you have. And so you are calling genotype. So this, for example, for this specific place, all the new sequencing result is all G. So that individual is GG for this specific position. For some other position, for example, so similarly, this is a reference genome, you map the reads, you find this position is all A, so you have AA at this position. For other position, you have C here. You have A in some of the sequencing result, you have C in other sequencing result. So for this position, is heterozygote. So by doing variant calling or genotype calling, you can call homozygote or heterozygote for the new sample. And then, so next concept I'm going to introduce is called haplotype phaging. But before we go into that, I want to give you a break, so you come back in 5 minutes. And the reason they need to do both is that this whole genome sequencing, they have very low depth. They don't know how accurate technology is, so they use a whole genome sequencing for them to check the quality. So basically, the idea is that for the exome sequencing, and even for the genotyping, for some specific condition, the genotyping is considered the technology to be most accurate. Exome sequencing will be more accurate than whole genome sequencing. So you have these two. This is the golden reference. You don't, whatever it is found with this technology, they're accurate. And then you can compare those two technologies to this one, and then compare whole genome sequencing to whole exome sequencing. And you need to, because when you do the analysis, when you do mapping, there's a lot of technical details about how stringent you are. You want to place a read in a specific place. Do you allow one difference, or you allow two difference, allow three difference? So those are details, and those parameters sometimes is not intuitive at the very beginning. What you can do is that you try different parameters, you compare to a higher quality result, and then go back and to adjust the parameters. So they use different technology to help them get accurate, get a set of genetic variant and a specific accuracy levels. And there are always some level of inaccuracy or error in sequencing result, because no sequencing result is 100% accurate at the moment. And the other thing is that you can notice that some of the population they collect, for example, for the American sample, they actually ideally they could include some indigenous Native Americans. They can provide even more information about some local population, and also some part of the human population was not covered, and it was mainly some one of the problem was the constraint of the access to the population. And for Native American, some Native American population actually they prohibit the study of DNA in the population. They don't want people to use their DNA for genetic study. And the other consideration is just the availability of sample and the availability of financial resources. You cannot sequence too many at that moment, and then try to find populations that are representative of a specific region. But after this project, there are a lot of sequencing effort going on, and we have a lot. Right now, I believe we already have genomes that are representative of different populations. And in terms of how the pattern change if we include more population, I would say it won't change much in terms of the average difference across any two genomes. But if we sequence more human population, for sure we are going to find more mutation or more genetic difference. But those differences are likely to be only restricted to that specific population. We can talk about the effect of population on, especially different population will have different pattern of genetic variation. So we're going to talk about that later. So the concept I want to talk about is called haplotype phaging. And after you do a variant calling, the data you get, the result you get is genome. And basically, the places that is homozygote, but different from the reference genome, or the positions that are heterozygotes. The things that we know that, like, for each individual, we are different. And for example, for those two differences, you have AC at this position, you have CT at this specific position. How do we assign those alleles to the two chromosomes? And by assigning alleles at each variant position to a specific chromosome, basically you are going to have only 2 chromosomes for each individual. And this is called haplotype phasing. Haplotype basically is the combination of all alleles on a chromosome, so this is called a haplotype. Even if you have only 2 positions, AT is still considered as a haplotype. And similarly, this is also called as an allele. So allele can be used in many contexts. And identifying haplotype has a lot of implications and applications later. But here I just want to introduce the concept because it is used in the Thousand Genomes project. We don't actually talk much about haplotype in this specific lecture. Okay, so this is I would say the most important information for this lecture, relevant to this lecture, is the total number of genetic variation if we sequence 2,000 individuals. And I think I just want you to pay attention to the last column, because with different technology and looking at different regions, you're going to have different numbers. So in total, we have about 88 million genetic variants identified in 2,500 individuals. And they also have about 3 million indel. And one interesting thing for you to notice is when you look at the length of indel, so indel is insertion deletion polymorphism, so we can determine how long is the insertion and how long is the deletion. And on average, it's only about 3 base pair long. So most of the indel they found with this technology are very short. And there are other numbers for different type of variant, structural variants, and multi-allelic site, multi-allelic SNP. You can see that bi-allelic SNP is much more common than multi-allelic SNP. SNP, single nucleotide polymorphism, is created by point mutation. So if you have 1 point mutation, you have 2 alleles. So it's bi-allelic. In order to have multi-allelic SNPs, you require mutation to happen at the same position at least twice. And it's not very common in reality. And I just want one thing to take away from this figure is that the scale of the number of differences we can find in the human genome is about 88 million. And a more interesting information is what about the difference in a typical genome? Like if we get a genome from a new individual and then compare the genome to the reference genome, how many differences am I going to find? And the number is slightly different across different human population, but overall for SNP, in African it's like 4.3 million. In South Asia it's about 3.6 million. The scale is about 3.5 to 5 million single variant we can find in a new genome. So this is the number I was talking about. It's about if we compare a genome to a reference genome, typically you can find about 4 to 5 million variants. So remember that the variant we are talking about here, it could be homozygote of alleles. They are different from the reference genome, or it could be heterozygote. And so for those genetic difference we find in the new genome, 99% of those differences are very small difference. It's either difference at 1 nucleotide or a few base pair, a few base pair segments. Only a small number of those variants are structure variant, and you can look at different kind of structural variant, deletion, insertions, inversions, and copy number variations. The specifics of different types is not important. Don't try to remember that. Just remember that we have a very small number of structural variation in our genome, a huge amount of small single nucleotide or insertion deletion polymorphisms. The interesting thing is that for structural variation, although they only happen at about 2,000 places in the genome, but because the structural variation is low, it involves a huge amount of nucleotides, the total amount of base pairs that are influenced by structural variation is actually huge. It's 20 million. It's actually much more than the SNP and indels. So structural variation, although not a lot, but if they happen, they can have a huge, a big impact in a specific context. And so this is basically the information from the table, but the total number of variants per genome, and you list them, plot them by human population. And here are the European population, here are the East Asian, South Asian, American population, and these are the African populations. And apparently, one apparent trend is that the African population, like African individuals in the genome, they tend to carry many more genetic variants. And for the American population, the individuals they collect at American, that has ethnic ancestry from African, European, Native American, you can see that the number vary a lot. It's actually the group that has the highest variation. And the variation they found here is actually associated with how many African ancestry they have. Basically, for individuals that has more African ancestry, they tend to have more genetic variant. And basically, that's because that is influenced by the African ancestry. And
you might be getting to this, so if I'm jumping the gun, I apologize.
No,
but why is that African ancestry interviews have greater variance?
Yeah, that's a great question, and it's a very important piece of information scientists use to decipher our human history. Basically, where do modern humans originate, and how do we migrate and occupy different places of this planet? And based on the current theory, modern humans originated from Africa. Especially our ancestor was there, and then a small group of those our ancestors go out to Africa and then travel to different parts of this planet. So think about that. We have a pool of genetic variants at the very beginning, and only a few of them, a subset of those individuals, they go out of Africa. So they only carry a subset of those genetic variants. And that's why the African individuals, they reserve a huge amount of genetic variations. So this basically this pattern is consistent with the model we have right now about human origins. And later we have a whole lecture dedicated to human history, like where do modern humans originated, how do we migrate, and how do people mix, how do different populations mix. And or we can make this kind of inference about human history using the genetic variation data we get. And so the number we have here is basically the number of variants we observe in each genome. But when we look at so for each variant, we can ask how often is that variant observed in the total sample we have. And some variants are very common, like you have many individuals that carry that variant. Some variant is very rare, only one individual or one chromosome in one individual have that variant. So that's the frequency of that specific variant. And so here I use reference allele. Basically, it's the allele in the reference genome, and the alternative allele is the non-reference allele. And the frequency we're looking at here, because at each genetic variant we have two possible alleles, and we only look at the frequency of the alternative alleles. And remember that the frequent for most of the prediction, it has only 2 alleles, so the frequency of those 2 added together will be 1. So most of the genetic variant has very, very low frequency. So in total, we have about 80. So remember that we have 88 million genetic variants in this whole sample, 2,500 individuals. Only 84 million of them are biallelic, so you only look at those variants. 64 million of them, is about 3/4 of those variants, has very, very low frequency, less than 0.5%. Only a small number of them, is about 10% of those variants, has high frequency, higher than 0.05%. So basically, the majority of the variants we observe in this sample, in the human population, has very, very low frequency. They are very rare. So this is at the level of many samples. What about we look at the variant we observe in a single genome? So in a single genome, we know that there's about 5 million genetic variants, and we ask, are those variants common in the general population? And so out of the about 5 million variants in a single genome, the majority of them is quite common. So this figure lists the number of genetic variants observed in a single genome that is also has low frequency at the global population, and it's a small number. And basically, for any individuals, if we find a variant in the genome, we could be quite confident that that variant is also present in the population. And I have a concept here, singleton. Singleton basically is a genetic variant that is observed only once in a specific sample. And you can see that the singleton usually is a new mutation that creates a new allele. If a new mutation just happened, it's going to only influence one chromosome, so it's a singleton. And if you look at singleton, the number is relatively constant across human population. So human history doesn't influence the number of singleton, because singleton is new mutation. Mutations can happen most recently. So this is the sharing of genetic variant across population. The gray, darker gray, are the variants that share across all population. And basically, the idea is that for populations that are close to each other, they tend to share genetic variants. And most of the variants we found, because they are very rare, they tend to restrict to a local region. And so we were only talking about these, basically the number of genetic variants. And now we want to move on and talk about where are those variants in the genome? Are they in the gene, or are they in the non-coding sequence? And this is considered as the functional context of those variants. If those variants happen in a coding sequence or in a regulatory sequence, they are more likely to have some consequences. And here are basically more concepts about different type of mutation for the functional context. And here we only focus on single nucleotide variant, and they're created by point mutation. Sometimes a point mutation can change the codon, but those two codons, they code the same amino acid. So in that case, it didn't change anything in the protein sequence. So this kind of mutation is called silent mutation, or called synonymous mutation. Sometimes the mutation creates a subcodon. It's called nonsense mutation. And then if you change the amino acid, it's called missense, or non-synonymous mutation. And sometimes, even in the case that change to different amino acid, sometimes it can still reserve the same kind of chemical property. In the case, you didn't change that protein that much. Compared to this kind of case, you change the amino acid, you change a lot of the chemical property of the amino acid. In that case, you're likely to change the structure of the protein, and it's likely to have a stronger impact. Friendship mutation are more likely to happen by indel. So you have one insertion, you shift the whole radial frame. And protein truncating mutations are mutations that either create a stop codon, so you truncate the product, or you create some friendship or some large structural variant, mess up the whole protein coding sequence, and you either have no product or a shorter protein product. For a typical genome, it has about 150 protein truncating variants, 10,000 non-synonymous variants, but many more regulatory variants. So basically, because the protein truncating variants and amino acid changing variants are variants that happen in the coding region, and coding region is only 1%. So there are a small number of them happen in the coding regions. So we can envision that one very important aspect about genetic variant for human biology is that they change gene expression, or they change they regulate gene expression. And so for the last 2 slides, actually I don't need to spend too much time on those last 3 slides. For these 3 slides, I just give you an example of what you can do with genetic data. So right now, you have genetic data from 2,500 individuals from different human populations. One thing you can do is that you can use those genetic data to study the population structure of those populations. And for this specific analysis, you have different human population. You can ask one question. For example, if we assume we know that all human population come from one common ancestor, so you have k equals to 1. You can say ancestral population is 1. We can say that all population come from the same ancestor. If we assume we have two ancestor population, and then we ask for each individual what's the proportion of that individual has ancestry from one ancestor population. But when we set the ancestral population to 5, basically you can see that here are the East Asian population. So one of the ancestral population will correspond to the ancestral population of East Asia. And here are the South Asian. So the blue color represent the ancestral population of South Asian. And the green color here are the ancestral population of the Europeans. And here are the African population. You can see that they have a mixture of the European ancestry and the East Asian ancestry. And they have slightly ancestry that is common with the African population. So the number of ancestral population is arbitrary. You can set as many as you want. But doing this kind of analysis can help you understand how populations are closely related to each other. You can also infer the demographic history of each human population. For example, for African population, for any population, you can infer the population size of that specific population over history. And here you can see that here is the time from history to the recent time. And the scale is a little bit weird. You don't need to worry about that part. One important point is that in this specific historical time, all human population share the same history. Their ancestral population have the same population size. At this time point, the African population separated with all other non-African populations. And they all go through a reduction of population size at this specific period. In the most recent history, the population size increased dramatically. So basically, we can use genetic data to infer population history, and we have one lecture dedicated to this kind of analysis. Another thing you can do is to use genetic variation and try to find mutations that has very different frequency across the population, and that kind of difference could be a result of genetic adaptation. Genetic adaptation is that if you, for some human population, if they carry a specific mutation, and that mutation can help them survive better in a specific environment, that mutation will become more common over time in that population. For example, here, that yellow allele is 100% in Eskimo, but this yellow allele is about 20% in South Asia, and that's related to some genetic adaptation history we are going to talk about in one specific lecture. Another thing you can do is that now we have this large amount of genome for different human population. Basically, we create a reference data set, and we have data at the haplotype level. And now assume that we have some new data, we sequence some new genome, but because of some unknown reason, maybe because of lack of funding, we won't be able to do good enough sequencing. So we have some missing data for this specific position. And now we can computationally sequence this haplotype by looking at the reference data, because you can see that if you have a G allele and T allele here, this is always an A. You have CC allele here, this is always an T. So based on that kind of information, you can infer information for this genotype. So this is called genetic imputation. You can statistically infer missing data based on a reference data set. And the Thousand Genomes Project create this kind of reference data set. So that's what, and we are going to talk about this. We have one lecture dedicated to this. And that's what I want to cover for today. And we have 1 minute. We have 1 minute. Any questions? If not, oh yeah, there's one question.
So when there was like a decrease in population size, what was that event?
For this,
Yeah.